Fantastic, thank you. It's a real pleasure to have this chance to give this talk, and great to see so many people here on a Monday evening. So what I'll do is I'll tell you about our research, and in particular how we're bringing together approaches from the physical sciences and the biological sciences in order to try and understand at the molecular level the fundamental mechanisms that underpin both biological function and malfunction. So if we take a biological system and we zoom in, what we find is the fundamental unit of life, the living cell. But if we now ask the question, how does a cell work? The answer to that is through protein machinery. And so proteins typically, in order to be functional, have to interact with other proteins. And if you look at an animation like this, which captures actually roughly the correct density of biomolecules inside the cytoplasm of a living cell, it immediately raises the question of how is it possible for protein molecules to find the correct partner to interact with and avoid interacting with the incorrect partner. So understanding and discovering the fundamental principles which allow biology to assemble functional machinery out of this level of complexity, and indeed understanding what happens when this process fails, is really the sort of central theme in our research. Proteins, of course, traditionally have been studied in isolation, and we understand quite a lot about how proteins, for example, fold, how they go when they're freshly synthesized. They have these types of disordered states, but then they can undergo a transition into these folded states which have a well-defined shape. So we understand quite a lot about proteins as individual molecules. But proteins are very sociable molecules. They work together. They form larger scale assemblies, functional complexes, clusters, mesoscale phases. And it's becoming increasingly apparent that it's really the balance between these different states which defines the balance between function and malfunction in biology. And so what I'll do today is I will tell you about two of these states. So I'll be talking about the liquid condensate state of proteins. So these are small liquid droplets which we have pretty much in every single cell of our bodies, and they are functional, they're organized, they're sort of a housekeeping mechanism, they organize the molecules of a living cell, they bring things that need to be together, they bring them together. I will also talk about, so this is the sort of liquid state. I'll also talk about a solid aggregated state of proteins, the amyloid state. Typically when this state is formed, at least in uncontrolled ways inside our bodies, it is associated with disease rather than function. And it turns out that there's actually very close connections between these functional states and these states associated with disease. One of the most dramatic cases where this type of transition into the solid amyloid state takes place is actually in the context of Alzheimer's disease, where it's actually not just one but two proteins which undergo this transition. So there is the A-beta peptide, which is normally supposed to be in a soluble state, but during the disease development, onset and development, it forms solid aggregates in the extracellular space. And I'll tell you in just a moment about some of our work on understanding how this process happens for this A-beta peptide. It turns out that in Alzheimer's disease there's also a second protein which undergoes this type of transition from solution to solid, which is the tau protein, which forms solid aggregates now inside the cell. And these types of aggregates tend to be very difficult for cells to deal with. They're solid, they're intractable, they're difficult for the cell to get rid of, and therefore it makes, in particular, sensitive cells like neuronal cells rapidly very unhappy, and that's really what leads to ultimately neuronal death and loss of neuronal mass, and ultimately the symptoms associated with this disease. And it's sort of interesting to highlight the scale of this. So if we just take the example of dementias, of which Alzheimer's disease is the most common example, for about the past 10 years or so, it's been one of the main, if not the main, cause of death here in the UK, and it's almost comparable to all forms of cancers added together. It's a very, very significant problem, and at a fundamental level reflects the fact that even though we understand increasingly well the properties of individual proteins, we understand comparatively little about what causes them to assemble into different types of structures, either ones which are functional, or in this case, ones which are associated with disease. But of course, this is the level at which we have to understand these systems. We have to understand them at the, really at the supramolecular level, at the systems level, rather than staying at the individual molecule level if we want to really get to the bottom of biological function and malfunction. But of course, one of the features that is very apparent when we go from individual molecules to molecular complexes and molecular systems is that the complexity increases very dramatically. So one of the big themes in our laboratory here is trying to think of new ways of generating data, new ways of understanding data which allows us to access this system scale. And again, it's important to highlight that many of the key characteristics which underpin function and malfunction are only apparent when you work at the system level. So again, if we go back to the example of this A-beta peptide which aggregates in Alzheimer's disease, the peptide itself is definitely not toxic. So in fact, in the central nervous system of all of us here, we have this A-beta peptide. It's definitely not toxic. So this toxicity only emerges when it associates with itself in the incorrect manner. So it's really at this sort of systems level. And something very similar takes place with functional systems. So many of the characteristics that make biomolecular condensates helpful for the functionality of the cell, they only emerge, the emergent properties, they're only there when you look at these molecules at the system level. So this is all very exciting. So we're really trying to move protein science from looking at individual molecules to molecular systems. The challenge, however, is that by doing so, we rapidly end up with systems which are just too large and too heterogeneous to be able to be studied easily using conventional methods which work fantastically well with individual proteins. Also, even though these systems are larger, they're typically still much smaller than the wavelength of light, this diffraction limit here, which means that we can't easily look at them using optical microscopy. So they fall really into this type of physical length scale where many of the existing methods struggle. So a big part of what we do is really developing new ways of studying these systems. And this is one big direction of travel in physical sciences in general, is trying to push the boundaries of what we can measure, how complex systems we can understand. And the way I like to look at this is in terms of size and scale. So if we look at what's happening in chemistry and biology over the years, we've really gone from doing chemistry on scales of liters, in a sense. That's sort of a human scale. That's something, a flask you can hold in your hand, to obviously test tubes, which are more economical in terms of material. And what will currently be, in some sense, state of the art in academic labs like labs here, but also in industry, would be multi-well plates and maybe robots using these multi-well plates. And of course, the further you go into smaller volumes, the more data you can generate for a given amount of time and a given cost. And so that's why there's been this long-standing trend. It turns out, however, that somewhere around here there's a fundamental limit. So it turns out that you can't easily, for example, pipette liquids of the surface tension of water on volumes which are much smaller than a microliter. So if we want to continue this trend towards higher content measurements, what we have to do is we have to integrate these fluid manipulations onto a device, onto a microchip. And so this is really something that we're very actively pursuing here. So these types of microfluidic devices, drop-off microfluidic devices, I'll come back to these. They're really the next stage in this type of evolution. And these types of systems have actually already transformed many areas of biology. So the single-cell transcriptomics revolution was really powered by drop-off microfluidics. And what we're finding is these types of techniques have, I think, at least the same kind of potential for understanding proteins. And so as we go along, I'll highlight how increasingly by doing experiments really with these types of cutting-edge techniques on small scales, we can generate more data, and we can, in some cases, do experiments which would be very hard to do on large scales. And it's interesting, these types of trends of size and scale are actually apparent in many other areas. So this was one analogy that I think was first pointed out by Steve Quake, which is that actually very, very notably, of course, in the microelectronics revolution has been a very similar kind of trend really where effectively, if we go from vacuum tubes to solid-state transistors, to integrated circuits, to microprocessors, what's happened is that actually the microprocessors that we use today, they're nothing else than a collection of transistors, and they're still moving electric currents around. What's changed is the level of integration and scale. However, these objects here on the right-hand side, they can solve qualitatively different problems. So with a microprocessor, you can solve a qualitatively different problem than you can with a single transistor. So scale and integration is really important when we think about measurement in physical science. So what I'll do here is I'll show you 3 examples from our research. So I'll first talk about the mechanisms by which the proteins can transition from soluble states into aggregated states. How does that process happen? How can we understand that process? I'll then talk a little bit about functional systems, these bimolecular condensates, but I'll also highlight that there's a very strong connection between these functional systems and disease-associated self-assembly. And then hopefully I will have time to show you some examples of how we're using our understanding of protein self-assembly in order to generate new types of materials with natural building blocks, but we're reassembling them in new ways to generate technologically interesting materials. Okay, so let's start with aggregation. So as I mentioned just a moment ago, there's a number of diseases actually which are connected with the transition of proteins from soluble states into aggregated states, Alzheimer's disease being one of the most prevalent ones. And what we wanted to do is we wanted to try and see if we could actually understand the mechanism by which this process happens. So how and why is it that proteins go from being soluble to generating these types of structures? And so the way we do this is we bring together, again, physical sciences and biological sciences in order to generate this type of mechanistic understanding. And so the experiments that we carry out in our lab look something like this. We can actually recapitulate this aggregation process in the test tube. And I'm showing this again on the basis of the A-beta peptide, which is the peptide which aggregates in the extracellular space in the context of Alzheimer's disease. And so this is what the experiments look like. So we start off with all of the peptide is in its soluble form. And then, as a function of time, it ultimately converts all into the aggregated form here. This was at low concentration. If we increase the concentration, what happens is this reaction takes place more rapidly, as one might expect. But now what we can do is we can bring in sort of the physical side of things, and we can do some modeling, and we can ask, well, what type of mechanisms might describe this process? So the simplest mechanism you could think of is one which looks like this, where effectively we have the molecules which simply clump together and form an aggregate, and then this aggregate can grow. And this is what's known as primary nucleation. And the lines here correspond to the mechanism, and the data points are the actual experiment. And you can see it doesn't really describe the experiment fully. So it looks like the aggregates of this peptide associated with Alzheimer's disease, it looks like they don't form in this sort of simple way by molecules just clumping together. Something else must be happening. So what could that be? Well, we thought about this. We said, well, maybe what happens is maybe the aggregates, maybe there's one or two of them that form, and then they start fragmenting. And because they grow from the ends, then this would accelerate the growth process, and they might be multiplying in that way. And so again, the nice thing about these modeling techniques is we can test this mechanism explicitly. So what happens if we now take this type of model and we test that based on the data? You can see the lines here still don't match the data. So interesting. So this peptide, the aggregates, they're not forming from just peptide molecules clumping together. They're not forming by existing aggregates fragmenting. So the only physically meaningful possibility that's left is it has to somehow involve both. It has to involve the healthy form of the peptide. It has to involve an interaction between the healthy, normal form of the peptide and the aggregated, disease-associated form. That's the only thing that's possibly physically meaningfully left. And so if we do that, then effectively we find that we can actually describe this process in very good detail. And this process is known as secondary nucleation. So we originally discovered the role of the secondary nucleation process in the aggregation of A-beta by using these types of techniques, but it's now been seen by many other groups using other techniques as well. And the reason why it's interesting is because it means that this aggregation process has some very specific and potentially actually dangerous characteristics. So what happens is the following. So initially we have these peptides which are in solution, and there's a very slow step where few of these aggregates form. So initially a slow step, and this is what we call primary nucleation. But as soon as we have even a small number of these aggregates present in the system, now of course this secondary nucleation process can take over. It turns out that these aggregates are dangerous in the sense that they can catalyze the formation of new daughter aggregates. So here I have one aggregate and a new one nucleates here on the side, and now I have two aggregates. But then of course each one of these can now in turn act as a source of new aggregates. And so effectively what happens is that there's a very rapid multiplication of these structures once they initially form. So if biology doesn't manage to stop this process at a very early stage, then it becomes increasingly difficult to control because there's more and more of these aggregates. So this was for one particular peptide. This was the A-beta peptide associated with Alzheimer's disease. But it turns out that there's over 50 different diseases which are associated with this exact same physical process, the transition of proteins from a soluble state into an aggregated state. So we then asked the question, do all of these other proteins have a similar type of mechanism, or is there something else that's happening? And so this is one way of looking at this system. So we can, from this type of analysis, from the modeling, we can get a doubling time. So we can ask the question, how long does it take, if you have one aggregate, how long does it take for that to generate a new aggregate and I have two aggregates? And so the answer to that is it's somewhere between hours to days, maybe a week. And the reason why there's a range here is because the concentration of this peptide varies from one person to another. So this is roughly the sort of range. And now we can ask, well, this doubling time here, is this something which is relevant on the timescale on which the disease develops? And so this is, I'd say the onset and development of the disease is on a timescale of about 10 years or so. And so it turns out that, of course, if the aggregates double even once a week, then this would be very significant. There will be more than enough time for this process to take over and really drive the development of the disease. So we can sort of draw a line here where things which replicate faster than this line, so on this side, this diagonal, those are the ones which are dangerous. So they have this sort of out-of-control multiplication property, and things which are below this line, those are ones which are in this sense, safe forms of assembly. So then what we did is we looked at a number of these systems, and this is a summary of many, many years of work which was led by a fantastically talented student and postdoc in the group, George Meisel. And so what happened and what we discovered by doing this was that all of the proteins that we looked at that were associated with protein aggregation diseases, they all seem to have this property that they underwent this self-multiplication process. So for example, there's alpha-synuclein, which I'll talk about in just a moment, which is associated with Parkinson's disease. There's tau, which is the other protein which aggregates in Alzheimer's disease. Prions, of course. IAPP, which is associated with some forms of metabolic disease. They all seem to have this property of undergoing uncontrolled multiplication. So that raises the question, well, is there anything here at the bottom of this type of phase space? And it turns out there are things. So for example, actin, which is the basis for the cytoskeleton of the cell, that obviously doesn't have this property, and you can imagine that wouldn't work very well if you wanted to build a sort of cytoskeleton that's sort of like scaffolding for the cell. That wouldn't work very well if the scaffold started to multiply in uncontrolled ways. So that makes sense. But what's interesting is there's many structurally related forms to these disease-related forms here. There's even functional amyloid systems here, which are found in this bottom right-hand side. But the interesting thing is none of these are associated with disease. These are all materials which are used by nature for functional purposes. So it looks like in cases where nature requires these types of structures to be formed, it seems to have out-evolved this dangerous property for self-multiplication. So we then thought, well, if this is the characteristic of disease-related self-assembly, maybe we could try and explore this more broadly as the basis for biomarkers. And one of the big challenges in this whole space is that there's no early detection approaches for any of these diseases. So typically by the time that people get diagnosed, it's very, very late, and it's very hard to do anything about these diseases. So I'll show you some of our efforts where we've been trying to use some of these concepts to try and develop new types of biomarkers. And I'll show you this on the basis of Parkinson's disease. So this is in terms of the physical chemistry, a very similar type of process. The protein is different. So the protein here is called alpha-synuclein, has natural roles in synaptic trafficking, so how neurons talk to each other. But during the onset and development of disease, what happens is it undergoes a transition to form these types of aggregates, and then in a very similar process to what I just outlined, the aggregates can multiply, and then they end up as these large-scale aggregates, clumps inside neuronal cells, and in Parkinson's disease, these are known as Lewy bodies. And again, it's very hard for the neural cells to deal with these aggregates to really clog up the functioning of the cells and make the cells very unhappy and ultimately result in cell death. What's interesting is these aggregates can actually even be transmitted from one cell to another. And so what that means is there must be, at least transiently, small amounts of these misfolded, dangerous, aggregated forms of the protein, which must be circulating in the body. So we thought if we had a way of really sensitively detecting them, that would give us a way to identify these types of disease states, hopefully at an early stage. So how do we do that? They're obviously present in very small quantities. We have to be able to distinguish this disease form of the protein from the healthy form here. And so what we decided to do is to exploit this property that they are able to self-multiply. So if we can extract even a small number, but we can then, in our assay, get them to multiply, then that will give us a better chance to actually detect them. So this is the idea. And so these are known as seed amplification assays. It turns out that this is a relatively challenging experiment. There's very low concentrations of these species. So it's very hard to actually determine their level. And so the way we've approached this is by, again, coming back to these ideas of size and scale. So we said, what happens if instead of running one of these seed amplification assays, where if you put one of these seeds together with the precursor healthy form of the monomer, then obviously the seed will multiply. So that's why it has this kind of, this is what's known as the seeding property. But we said instead of carrying out one seeding reaction, what happens if we now carry out thousands of these types of reactions in very small volumes? And in particular, if we make the volumes so small that we're sure that each one of these compartments contains either 0 seeds or one seed. So if we do that, at least in theory, this becomes a very simple experiment, because if we want to know how many aggregates, how many seeds we had initially in this test tube, the only thing we need to do is split it up into lots and lots of small compartments, carry out lots and lots of seeding experiments, and then just go and count how many of these compartments were positive. So it really becomes a digital assay, which is why we call this digital seed amplification assay. So for example, this compartment here didn't have a seed in it, it's negative, and this one here had one seed in it, it was positive. And then if we have a higher original concentration here, then we'll find more of these compartments which will become positive. So that's the idea. So how do we generate large numbers of very small compartments? And again, this is where we turn to these integrated droplet microfluidic techniques. So this is what the devices look like. So we make them here in the chemistry department in our laboratory. The characteristic length scales are of the order of micrometers. So this scale bar here is 200 microns. That's roughly the size of a human hair. This video is slowed down roughly by a factor of 1,000. So this happens really at very, very high rates. And so the idea is each one of these droplets, the clinical sample comes from here. We bring in the soluble healthy form of the protein from this side here. And so each one of these small droplets contains either 0 seeds or 1 seed, and in all cases lots of healthy monomer. And then the only thing we have to do is to go and look into these, we incubate these droplets for several hours, and then we go and ask, well, was there amplification or not? And the way we do the readout is we have a optical setup here where we can interrogate each one of these droplets in turn. And so this is what the data look like. So we have a signal here, which we call the gating signal, which lights up every time there's a droplet that goes by, just so we know a droplet went by. And then what we do is we have another signal here, which only gives us a positive signal if we have aggregates present. We have a special type of dye which only lights up in the presence of aggregates. And so you can see here, this droplet here, we know there was a droplet because there was a spike here, and this one gave a signal in the aggregate channel. So this was a 1, this was a positive, there was something there. The next one, the droplet lights up, but there's nothing in the aggregate channel. The one after, droplet goes by, nothing in the aggregate channel. So by doing this, we simply go in and count how many of these droplets were positive. And so the way we do this now is we get patient samples. So we've been doing a lot of work with cerebrospinal fluid, which you can actually get relatively readily from lumbar puncture. And then what we do is we take this sample and we encapsulate it into droplets. And this is what healthy people look like. So almost all of the droplets are empty. So we're histogramming here the intensity of the spike. So almost all of the droplets are empty. But if you now do this experiment with somebody who has been diagnosed with Parkinson's disease, it looks very different actually. So you take this sample, and again, most of the droplets are still empty. So most of the protein present is in the healthy form, but you start to see here this second population, where effectively we had this seeding activity, so there's droplets which have had seeds in them, and this corresponds to the misfolded aggregated form of the protein. So on an individual patient basis, this is what it looks like. So here we looked at 20 different patients, and this was actually initially done in a double-blinded way. So we didn't know which ones were which. But if you do the measurements, what's interesting is they clearly seem to fall into two categories. So most of the samples on this side seem to have a very low concentration of seeds, whereas there's a group here which has a high concentration of seeds. And so then it seems natural to say, well, our prediction would be these are the ones which have Parkinson's disease. And then if we do the unblinding, then effectively that turns out exactly to be the case. So with this type of approach, I think there were 19 out of 20 which were identified correctly, and it gives us this very good performance here in this assay. So that's interesting, because having a way to identify people with Parkinson's disease based on quantitative aggregate level measurements is something which is very, very hard to do at the moment. So we're excited by the fact that we now have an approach to do that. But what's interesting as well is that these types of digital microfluidic measurements, they quantify the number, the absolute number of these aggregates. And so it seems that there's really a threshold value here, where as long as you don't have aggregates which are at a higher concentration than this threshold, then biology seems to be able to deal with them and you don't get the disease. Because even if you look at these people here who do not have Parkinson's disease, the number of aggregates is not exactly zero. So with these very sensitive methods, we can still detect aggregates. It just doesn't seem to cause problems until you hit this type of threshold here. And this is where it becomes difficult for biology to deal with this problem. This is really where disease onset takes place. So this is one example. This is one example of an approach where by going down in scale and up in terms of the level of data that we generate, we can actually understand systems which would be hard to look at using conventional techniques. I'll now say a couple of words about the liquid condensate state of proteins. So this is a different state from the solid aggregated state that I've spoken about so far. These are typically functional structures. They're liquid droplet-like structures inside living cells. In fact, they exist in pretty much every cell of our bodies. They carry out various roles. So here's a couple of examples. So they can be involved in transporting nucleic acids from one side of the cell to another. They're involved in the stress response. So if cells get stressed, they have to rapidly move molecules around, sequester some molecules, in particular nucleic acids. And so biology does that with these types of stress granules, which is a name for these condensates. And even things like the synthesis of some of the very fundamental building blocks of the cell. For example, the ribosome, that's the machinery that makes proteins inside living cells. And so if you ask the question, well, where's the ribosome made? It turns out it's made in a particular subcompartment of the nucleus. But in order to keep this process sort of well localized, so it doesn't mess up everything else that's going on inside the nucleus, what biology has done is use this type of small liquid droplet here, which is known as a nucleolus, as the site of the ribosome biogenesis. So there's many examples of these functional systems in biology, and it's really a housekeeping mechanism helps keep molecules in the right place at the right time. However, it can also be dangerous, because by bringing these molecules together, what the cell does is it creates more opportunities for molecules to meet each other, and some of those interactions may lead to problems. And so the way we originally discovered about this was actually not by thinking so much about condensates, but actually asking the question, when we see these disease-related aggregates, what happens before the aggregates appear? So I'll show you here a video which we generated over 10 years ago now, where what we did is we we asked this question on a specific core recognition motif of this A-beta peptide. And so what we saw, this is again under high magnification in one of these microfluidic devices, so there's no convection, only diffusion. And you can see here the aggregates appearing. You can see here solid phase appearing. You can see the fiber-looking aggregates. But what we found is before these aggregates formed, which ultimately take over the whole system, we saw these sort of round, liquid droplet-looking things. And when we looked at them slightly more closely at a later stage, we saw that they had all the properties of liquids. They have, for example, surface tension. If you put two droplets next to each other, they merge because of surface tension. So it really looked like these aggregates, the initial step by which they formed, it looked like it was really related to this liquid state. And so then we thought, well, okay, if these liquid structures are prone to undergo this transition into a solid state, the solid state must be therefore more stable. So maybe if we take one of these liquid condensates and we shake it a little bit, let's see if that makes it go into the aggregated form. This is true for all systems. So what we did exactly that. So how do you, in some sense, shake or shear an individual microscale droplet? That sounds like a hard experiment, but actually if you use these types of microfluidic techniques, it's a relatively more straightforward experiment. So we made ourselves one of these microfluidic devices where we trap an individual condensate here. And then what we're going to do is we're going to squeeze it into a channel on the right-hand side. So let's squeeze it in. So we're applying a bit of pressure. And you can see it squeezes in, it flows like a liquid. But if I now let go, you can see what happens. It springs back. So let's play that again. So the droplet is clearly liquid, so it flows. It looks like, small droplet of honey, say. It's a viscous liquid, it flows. But then if you let go, it bounces back. So liquids don't do that. At least when I put honey on my bread, that's not what happens. And so this type of behavior is clearly characteristic of solid. So it's elasticity. It looks like it's rubbery. So somehow between the beginning and the end of this experiment, this droplet has gone from being purely liquid to developing sort of low-frequency elastic modulus, which is a characteristic of solids. And if you now ask the question, well, how about some other characteristics of solids? Of course, what happens with liquids is the molecules inside the liquid are able to diffuse around very rapidly. We can see this by making the molecules fluorescent, and then we remove the fluorescence by photobleaching in the middle of this droplet. And then if we wait, then the molecules can reorganize and diffuse around, and it looks the same as before. But if we do the exact same experiment, but we now just apply a little bit of shear to this droplet before we carry out this type of experiment, what we see is this ability of the molecules to reorganize has completely disappeared. So now the molecules are fixed in their positions, exactly as you'd expect from a solid. So these types of systems are really prone to undergo this type of transition. And so we know that this is now the case for many of these proteins which are associated with disease. The early nucleation step in many cases takes place from a liquid precursor. It has been discovered that this is what happens for alpha-synuclein, this is what happens for tau. We've recently discovered that this is what happens even for the full-length A-beta peptide associated in Alzheimer's disease. The original slow nucleation step in many cases takes place from one of these liquid precursors. So based on these examples, I think it's sort of becoming clear that it's actually a very fruitful way of thinking about the different states that proteins can occupy, is really thinking about them as states of matter in the same way as you would think about a small molecule, for example, water, having different physical states. So it could be in a solid state, it could be in a liquid state, it could be in a gas state. And so if we want to understand the transitions between different states of small molecules, what we do is we generate a phase diagram. So what that means is we look at some characteristics, for example, pressure and temperature, and we ask the question, if we vary these properties, what is the state of the system? And then what we'll find is at low temperature, water obviously forms ice. If we heat, it goes liquid, and if we heat even more, we get water vapor. So what we want really is an analogous way of understanding the behavior of biomolecules. The problem with that is that typically for biomolecules, the relevant parameters are not so much pressure, sometimes temperature, but it's typically composition. So there's something different about the components in these systems. And so what that means is in order to generate these types of phase diagrams, we have to make lots and lots of copies of the system where we vary the composition slightly. And so again, how do we do this in a scalable way? This is again where we turn to these integrated high throughput, low volume experiments. And the experiments look something like this, a very similar sort of physical setup to the one we just looked at, where if we want to understand under what conditions these two components here form a dense liquid and under what conditions do they form a dilute solution phase, we just mix them in different proportions. And again, video slowed down by a factor of 1,000, so we're generating thousands of droplets per second, where each one of them has a slightly different composition. I'm illustrating this with this particular protein here, G3BP1, which is one of the structural components of the stress granule. So it's a protein which functionally forms liquid droplets with RNA molecules when the cell is stressed. And so then what we do is we look at these small microdroplets under a microscope, and we can see under some conditions the proteins inside have formed these small condensates. So you can see this droplet here has lots of small condensates. In this droplet here, we're under conditions where the solution phase is stable and there's no condensates. And then what we do is we have a convolutional neural network which looks at these droplets and classifies them into either two-phase or one-phase. Now, one problem we have is all of the droplets look exactly the same. So how can we remember what was inside each one of these droplets? And so the way we do this is we have to barcode them in some way. And this is what it looks like in practice. This is, by the way, roughly close to sort of real speed. So this happens really at very high speeds. So we look at each one of these droplets, and we've added a small amount of dye, which is proportional to the amount of a given component that we've added. And so that allows us for each one of these droplets to figure out what we put inside it. And so now, if we do this, we use this information here to remember what's inside these droplets, which gives us the ability to place the droplets on this kind of plot. And then we color them depending on which phase they are in. You can see a very, very sharp phase boundary. So the droplets go from being homogeneous to having these condensates as soon as we cross the threshold concentration. So this is a very sharp phase boundary in a very similar way to when you, have a water droplet, which, for example, goes solid. That's a phase transition. So this is a phase boundary in a very similar sense. So with this type of technology, you can do lots of different things. So now we can routinely generate phase diagrams. We can understand different aspects of the behavior of these systems. I'll just show you one example where we've used this to ask, I think, a really interesting question, which is, can we find small molecules, drug-like small molecules, which would change the phase behavior of these proteins? And their motivation, of course, is to try and find molecules which would rescue aberrant phase behavior when it takes place in disease. And so we've done this for many different systems. I'll show you some relatively recent data on associated with ALS, which is actually the most common form of motor neuron disease. So what happens in these diseases is again, from a physical chemistry point of view, very similar to what we saw earlier. There's aggregates formed inside neuronal cells. In this case, it's slightly different proteins. So the data I'll be showing you is based on this protein FUS. And so, of course, once the proteins are aggregated into solid structures, these tend to be very intractable. And so it's been incredibly hard for any of these diseases to find small molecule drugs which reverse the process once the solid aggregates have formed. So we thought, well, if many of these aggregates, and this protein FUS is one example, if many of these aggregates are formed from liquid precursors, maybe if we reduce the number of these liquid condensates before they have the time to go solid, maybe that's a slightly easier task, and that will result in us having less aggregates in the final system. So that was the idea. So how do we find small molecules which change the phase behavior of proteins? Well, with these types of microfluidic techniques, that's actually, at least conceptually, a relatively simple experiment. What we do is we generate phase diagrams. So this is an example of a phase diagram. This has a couple of thousand different points. Again, each one of these points is an individual measurement where we've made one microdroplet with a specific composition, and we've asked, do we have, inside this droplet, do we have FUS condensates, in which case it's colored red, and if we don't, then the point is colored blue. And now what we do is we take a small or big library of small molecules, which are drug-like. We've looked at different libraries, large libraries, hundreds of thousands of molecules, smaller libraries. I'll show you one example here with 1,000 different molecules. And now for each one of these molecules, what we do is we ask the question, does the phase diagram look the same or different when we add the molecule? And so the answer is, for most of them, it looks pretty much the same. So if you look at this kind of histogram here, most of the molecules, it looks exactly the same. They don't do anything very much. But every now and again, we find interesting molecules. For example, this one here, This one really changed the phase diagram very dramatically. So if you compare with this one here, there was a very sharp phase boundary here. It pushed the phase boundary all the way up here. So it's really dissolved many of the condensates. So this is something which is very hard to do once the structures go solid, but it seems like at the level where they're still liquid, we can with small molecules shift the phase boundary around. And interestingly, by the way, you can also find molecules which do the opposite. So these ones here actually make the condensates form even more readily. So we're not. Those are interesting for other reasons, but we're not looking at those with the view of looking at the sort of drug potential here. Okay, so this is how we can find a molecule. So let's look at one of them. So here, this is one which, again, if we look at the slightly more detailed analysis here, the phase diagram, you can see in red is the part of the phase space where we have these condensates, blue is the part where they're dissolved. If we add the molecule, micromolar concentrations of the molecule, you can see all of the condensates disappear very rapidly. So this is a test tube experiment. What happens in a living cell? So we can take these molecules. It turns out that when we looked at about 1,000 small molecules, we found 17 which had interesting effects on the phase behavior, and a good fraction of them actually still did the same in living cells. So here you can see the same process happening in a living cell. We've tagged the protein FUS here with a fluorophore so we can see its localization. And once we add even relatively modest concentrations of this drug, single-digit micromolar, we can see that these condensates dissolve. And what's interesting here is because we found this molecule from a screen where we looked at the protein condensate. So we know that it's hitting the target. One of the challenges with these condensate systems is they're very coupled to many other things that happen in the cell. So if we just looked at the cell level, it would have been very hard to know what these molecules do. Do they hit the target? Do they do something else? But here, because we have this ability to generate the molecular phase diagrams, we know that these molecules directly interact with the condensate-forming protein. Okay, so the work in living cells. Let's try and increase the level of complexity even further. What happens in an animal? So this work done together with Peter St. George Hislop's group, and also together with a company that was set up here in Cambridge, Transition Bio, where we looked at a worm model of ALS. So it turns out that you can express this protein inside worms. It forms aggregates in the same way as it does in humans. And so you can see here, what we've done actually very specifically is we've taken a mutant form of this protein which is associated with a genetic form of ALS, and that's the protein that's actually expressed in these worms. And so the worms have lots of aggregates of this protein. If we now add this molecule, what happens is we see a very significant decrease here in the concentration of the aggregate. So it's clearly doing what we hoped it would be doing even in the animal. It's getting rid of the aggregates by destabilizing the precursor condensate state. We can even look at the behavior of these worms. So it turns out that these worms exhibit some of the sort of phenotypes associated with the disease. So if you have these aggregates, it makes the worms less able to wiggle around, and so we can count that. And so this is a healthy worm. It's happy. It's wiggling around. This is a worm which has this mutant protein which is associated with a genetic variant of ALS. It's significantly less happy. It has some phenotypes which you might associate with a sort of motor neuron malfunction. And then if we add our molecule here, you can see it even rescues some of the functionality. So it makes these very unhappy worms slightly happier, and this is statistically significant, and it makes them able to wiggle slightly more. And again, it doesn't do anything to the healthy worms. So this doesn't just generically make the worms move more. It's really very specific. It interacts with the target protein. So this is fantastic for worms which have ALS. Obviously, there's still a long way to go for taking this into human trials, but we're very optimistic that this type of approach of going beyond molecules to molecular systems will open up possibilities to address some diseases which have remained incredibly hard to address by focusing on interacting with individual molecules. So we're very optimistic about that, and we work very interactively with both big companies and small companies to try and take this forward. And on a technology sort of level, one thing to highlight is one of the reasons why we were able to find these molecules was because we had the ability to carry out a large number of measurements. We looked at each one of these molecules in our library, on the basis of the full phase diagram. And so if we think about this a little bit, this is a particularly nice phase diagram. This is around 100,000 points. And so by using these microscale techniques, this is relatively straightforward to acquire. It requires a single device, and we need about 10 microliters of volume of the solution, and we can do this sort of in a matter of seconds to minutes. If we wanted to do this using even conventional, what's currently cutting-edge technology, robotics, microwell plates, just this one phase diagram would require 1,000 plates, and it would require 1 liter volume of the protein solution. And so this obviously now becomes impractical if we want to do this as a basis for screening. We want to generate millions and millions of measurements point. This would be totally impractical. So I think what we're learning is the more complex the systems become, the more and more it becomes key to be able to work with these small volumes so we can generate more data, we can understand the systems under more different conditions. This really allows us to make progress in areas where it's otherwise been challenging. So the final thing I'd like to highlight about these condensates is we've seen a couple of examples now of systems which form these liquid droplets, different kind of proteins. And so at some stage we asked the question, well, what is it about these proteins that allows them to form these droplets, and why don't other proteins do that? And so we turn to AI-based methods. Of course, AI has been incredibly successful for understanding structured proteins, so how proteins fold, what the structure of individual proteins is. So we wanted to ask the question, what happens if we now use some of these methods and we try to understand, can they tell us something about how proteins interact with each other? And in particular for these systems, many of the proteins are actually highly disordered, so they don't have a well-defined rigid structure. So the way we did this is we built 2 different types of models. So we built a model where we had some physical features, so we added physical features that were from basic physical chemistry, are important for phase behavior, for example, things like size and charge, something about the complexity of the sequence, information content, channel entropy, things like that. And we trained a model. And then we trained another type of model where we took a language model approach, where we said, well, let's not put in any a priori rules. Let's just have a very general language model, and let's train it on all of the proteins in the human proteome, so it learns in some sense the language of human proteins, and let's then fine-tune that based on a data set of phase-separating proteins. And if we do that and we take a consensus between these 2 models, it turns out that we can actually learn to a very high level of accuracy the specific sequence features which allows proteins to phase separate. So in this type of plot here, obviously what we want is performance to go up rapidly, so that means we have a large fraction of correctly identified proteins. So we can really learn that, first of all, there are very specific rules, and secondly, we can learn them very readily using these types of techniques. We then said, well, let's increase the level of complexity slightly further. Let's ask the question, what happens if we do this in the context of all of the other molecules that might occur in a living cell? So previously I showed you just whether we can learn the rules of individual molecules, phase separating. Let's now do this in the presence of all the other molecules that occur in a living cell. And so if we did that, it turned out that now we could only predict a smaller fraction of the proteins that were found inside these condensates. So that's interesting. Only a small fraction of the proteins which are found inside these condensates in a cellular context are there because of basic physical chemistry, phase behavior type arguments. So what are the rest of the molecules doing there? Why are they there? Why are they found in this condensate? There must be some other reason. So we thought maybe they interact, maybe the ones which form condensates, maybe they just generically interact more with other proteins in the human body. So let's test that. So here we have 2 data sets. These are the molecules which are found inside the condensate. These are molecules which are not found inside the condensate. And we're now looking at all of the interactions that they have with all other proteins in the human body. And that didn't really seem to be a big difference. It wasn't just that these molecules somehow are promiscuous, they just like to interact with all other proteins. That's not what's happening. But if we now take the proteins which are inside the condensate and we ask, do they like to interact with other molecules which are also inside the condensate? Then there's a very significant enrichment. And if you do the same with the molecules which are the proteins which are not inside the condensate, you don't see this kind of effect. So it turns out in a biological context, there's actually 2 reasons why molecules might be inside one of these phases. So either it might be what's called a scaffold protein. So either it's really prone to phase separate. It's prone to undergo this type of transition to form the dense state. That's some fraction of the proteins. Or it's there because it likes interacting with some of the other components which are already in this standard state. And if you add this to the machine learning model, now suddenly you go from this type of performance, which only describes a very small number of these proteins, to be able to describe really the majority of the proteins which are found inside biomolecular condensates from this type of machine learning model. So we then deployed this model across the full human proteome, and we said, well, now we have an ability to understand which molecules should form condensates. Let's try and ask the question, what this looks like across the full human proteome. And this is what it looks like. And so it looks like there's some, there's sort of some fraction of proteins which do form condensates, not all of them by far. Some of them are really not typically found in condensates. But a sort of a tail of things here at very high predicted propensity to form condensates. So we said, well, let's have a look at that tail. What is actually there? And so we took, in a completely unbiased way, we took the top 4 predictions from this machine learning model. And then what we did is we looked at them in cell images. And you can see all of the top 4 predicted condensate-forming proteins. You can see all of them actually in reality. When you do the experiment, you look up the data. You can see these spots here. So each one of these is an individual condensate. And if you now look at the bottom 4 as a control, you look at the bottom 4. Then in fact none of them form condensates. So this is really interesting. So these types of machine learning models, which have been very successful at predicting structures of individual proteins, it looks like we can really adapt them to have a very good performance to really try and predict and indeed discover new types of proteins which are present in this type of condensate form. So we're very excited about this ability to sort of discover new biology, discover new interactions, and I think this will give us even more ways of intervening in these systems when the behavior goes wrong in the context of disease. Very good. So in the final 5 minutes, maybe I'd like to switch gears a little bit and tell you something about how we can use these types of ideas to design new types of materials. And so the idea here really is, at some stage we thought, well, if we understand the rules by which proteins self-assemble, then we ought to be able to design the conditions to form new types of materials, which maybe are not the ones that nature uses, but nevertheless we should be able to access them, and maybe some of them could be technologically interesting. And we're doing this in a number of contexts, but before we start, it might be sort of, you might say this is a very strange idea. Why would you want to make materials out of proteins? Very strange idea. Well, actually, it's maybe not so strange. So many of the sort of really interesting and exciting materials in nature are actually formed from proteins. So, for example, spider silk. It's weight for weight as strong as steel, but it's also incredibly elastic. And if we think about this from a sustainability point of view, for example, it's actually quite remarkable in terms of the process. So you have a spider, it eats one fly, and that gives it enough energy and raw material to form this incredibly high performance material in a process which happens at room temperature, no harmful solvents, and almost no waste. So these are clearly interesting materials. Many other applications. This one here is interesting. So this is a mussel, which obviously is supposed to be stuck to a rock. And if it's somehow it gets unstuck, it has a real problem. So that really shouldn't happen. So in order to ensure that that doesn't happen, the mussel has a special type of adhesive which glues it to the rock. And that's actually a technologically hard problem. So how do you make an adhesive which sets underwater and is resistant for several different years? And so the way nature does this, of course, is by using protein-based materials. And of course, we do use proteins as materials for many different technological applications. But the question we asked is, well, maybe we could try and, instead of limiting ourselves to cases where nature has already made proteins into interesting materials, maybe we could try and re-engineer the assembly states into other types of materials. And one of them, I'll show you some examples here, where we're focused on trying to assemble proteins into structures which look like silk. So silk, obviously, fantastic material. But how do you produce silk at scale? So you can't easily farm spiders. That doesn't work so well. It turns out they like eating each other, so that's not a very fruitful way forwards. But obviously, fantastic material. And if you ask the question from a physical chemistry point of view, why does it have such amazing performance? It turns out because the molecules are arranged in a very specific way. So they're arranged in these types of beta sheet nanocrystals, which are then tied together by this more amorphous structure. So we said, okay, well, why don't we try and see if we could take proteins which are completely unrelated to silk, let's say for the sake of argument, plant proteins. Can we take plant proteins but assemble them in ways which are exactly the same as in silk? So in a sense, can we make vegan silk? Is that possible? Okay, so it turns out from a physical chemistry point of view that actually these types of structures here are stabilized by interactions which are common to all proteins. So again, from a physical chemistry point of view, it ought to be possible, but the question is, can we find the right parts of the phase diagram to be able to do this? And this is again where understanding the phase space, understanding the phase diagrams of proteins is very helpful. So for several years, doing a very systematic sort of search, fantastic work done by Oyaka Kamada, who was a student at the time, and Mark Rodriguez Garcia, was a postdoc at the time here, really did an absolutely fantastic study on this, and really managed to find not just one set of conditions, but a whole pile of parameter space, where indeed you could take plant proteins and reassemble them to form exactly these types of beta sheet nanocrystals connected with amorphous regions. And indeed, if you look at these structures now under high-resolution electron microscope, they effectively look indistinguishable from silk. So if you looked at silk, it would look exactly the same, and you can see an individual beta sheet nanocrystal here. So these are the beta sheets, and the rest is this amorphous region. So it really looks, even under high-resolution microscopy, exactly the same as silk. How does this material perform? So it turns out that by doing this careful assembly and making plant proteins look like silk, we get relatively high material performance. So on this type of material performance plot here, much higher performance than sort of conventional plant-based protein materials, and it really approaches what you could call sort of high-grade engineering plastics. So that was interesting. So then we said, okay, well, these are materials which are purely plant-based. We haven't modified the molecules in any way. We just reassembled them in ways which don't occur in nature. This could be potentially interesting. You can make larger scale materials. It turns out you can make them heat seals. You can make plastic bags. So we said, well, maybe we should think seriously about trying to replace some of the materials that are currently based on non-degradable plastics from non-sustainable sources. Maybe we should try and think about using this type of physical chemistry approach to replace them with plant-based products. And so in order to do that, we set up a company here in Cambridge, Sampler, and Mark Rodriguez is really taking the lead in driving this development, really with the view of trying to develop practically usable materials which are plant-based but have the performance which is normally only associated with plastics. And so the question is, obviously, if we use these natural materials, we don't cross-link them, we would expect them to be fully degradable. And so is that the case? So this is a fantastically technical experiment that Mark did, where he took these materials and he buried them in his back garden, and then he looked at them a couple of times a day to see what happens. And so we have different materials here. So we have plastic bag, we have PLA, which is one of the sort of supposedly degradable substitutes for plastics, but which is maybe not as degradable as we thought. And this is the material here. So let's try and play that again. It degraded so quickly that we couldn't even see it. So what happens, you can see it's decomposing very rapidly, and after just a couple of days, it's completely disappeared without any trace. The worms, in particular, seem to love it. But obviously, there's been more quantitative tests, and it turns out, again, that exactly what you'd expect is the case. These materials are not cross-linked, so they're very highly degradable, and the building blocks are fully natural, so they go back into the ecosystem. And in particular, there's good marine degradability, which is a real challenge for many of these other materials. So what we're doing now is we're thinking about really trying to replace non-degradable materials in as many applications as possible. So these are some examples from Sampler, where you can see anything that looks like plastic here is not plastic. It's one of these plant-based films. This one here is very interesting. So this is actually a collaboration with Gusto, the sort of food ingredients company. And it turns out one of the main complaints from the customers is, why is everything wrapped in plastic? So of course they were very interested to try and see if we could find some alternatives. So this is soup ingredients which are packaged into one of these films. And so this one here, we made it such that it actually dissolves on contact with water, which is sort of interesting, because then you can make the film itself from food grade materials if you want. You can use food grade proteins. So then everything becomes food grade, and it's really sort of, in some sense, there's zero waste. So not only is the waste in principle degradable, but here the waste itself is actually a sort of part of the functionality. So I thought soup was a bit boring. So then I sort of said, well, maybe we could package something else, for example, coffee. That would be more interesting than I'd be interested. And so what happens is an example. You can do the same thing with coffee. This is a particularly interesting material which only dissolves if you pour hot water on it. I think this is a slightly early prototype. It doesn't dissolve immediately, but you can see it's sort of largely dissolved and release the cargo. I think it requires a bit of stirring. So you can see here, this is sort of really scientists making coffee with a spatula. But in principle, you sort of get the idea. So I think it's incredibly exciting now by engineering the assembly of these proteins. We can really generate fundamentally new types of materials which don't exist in nature, but the building blocks themselves are completely natural, plant-based, unmodified, cross-linked. And of course, in order for this actually to be relevant, what we have to do is we have to go and really replace existing products with degradable alternatives, and that's a whole sort of story of its own, which is now happening at Zampler. And I think the team have done a really fantastic job. So of course in the lab here, we work with quantities which are of the order of milligrams. That's the quantity that you use in the lab. But of course the relevant quantity for having products in the market space is tons. So that's 9 orders of magnitude scale up. And so that of course has been an absolutely fascinating journey. Very good. So this is what I wanted to share with you. So I think it's a very exciting time in protein science. We're really moving from looking at individual proteins to looking at sort of supramolecular systems, protein systems. And I think that will be the key for really unlocking an understanding of both biological function and malfunction. And finally, I'd like to thank the fantastic group of students and postdocs here in Cambridge, with whom it's a huge pleasure and indeed privilege to work on these exciting topics. A lot of this work has also been done in collaboration with people both here in Cambridge, but also worldwide. And we're very fortunate that there's a number of individuals and institutions who fund our research, and without that, obviously none of this would be possible. And once again, thank you very much for being here and for listening.